Currently, the North Pacific Gyre is home to a Texas-sized garbage patch of nothing but plastic and other floating debris. This problem has a solution, a long-term one: bacteria. What kind of bacteria? Flavobacteria. Certain types of these bacteria could be called the flava-flav when it comes to plastic, because for them, it's always flava time. And when I say flava time, I mean time to break up plastics and polymers into cellularly digestible monomers with the use of membrane-bound proteins called enzymes. Here's how it works. The plastic particles are absorbed by the bacterium, and they enter the cytoplasm only to be greeted by an enzyme which will break it up. These enzymes are collectively known as nyloneases, and they are created by the cell's genetic code expressed through protein-crafting molecules, which read the mRNA to create sequences of amino acids which, in this case, code for an enzyme. The plastic particles bind to the active site of the enzyme, and the enzyme breaks up the polymer into small monomers, which then the cell can digest and eventually use to create adenosine triphosphate, or ATP, which I'm sure you remember from freshman biology is the energy currency molecule of almost all living organisms. Okay, Mike, I know you're wondering, Pierce, this makes no sense. These bacteria probably couldn't even survive going from one piece of plastic debris to the next. Plus, this is a totally inefficient process with too many byproducts, and these bacteria wouldn't be able to break up the plastic faster than we are putting it in. Maybe you're right, Mike. Maybe you're right. But instead of being pessimistic, let's just add some enhancements to our super awesome plastic-eating bacteria instead of moping around about it. See, what these organisms need is motility, the ability to move around using an organelle which gets the job done, like a flagellum. But wait, Pierce, Flavobacterium don't have a flagellum, so you're out of luck there, buddy. Wrong. Technology creates solutions to all of our problems. See, ATP can't be created without the use of an ATP synthase molecule which, when bombarded with protons from the electron transport chain, not only creates ATP, but it rotates. It's like nature's outboard motor. Attach a long, flimsy rod to this thing and you have a flagella. If we isolate the genes of the flagellated prokaryotes, say, I don't know, Salmonella, and multiply it using the polymerase chain reaction, we can actually insert that gene into the genome of our sexy Flavobacterium. I'm not entirely sure how the insertion process works, but they did it with other microbes, so I don't see why it won't work here. The result is a motile plastic-eating bacteria, which would be significantly more efficient than the current bacteria used to eat nylon. Sure, there are other things that could be done to increase efficiency, but this is just an example of how we can use genetic engineering to make our environment a much better place to live.